Hi everyone, Roland Kays here from North Carolina State University and the North Carolina Museum of Natural Sciences, talking about the Snapshot USA analysis tool. In this video, we're going to be covering looking at species richness. So how many species were detected by an array of camera traps? Here we've got our map of camera trap data that we have available to us from the Snapshot USA program that you may have helped collect. Each one of these dots is a place where we have sampled camera traps. And we can change the map if we want to look at it like this. If you click near, it'll tell you the name of the array. And we're going to change this to species richness. And now that, in this case, the dots are actually somewhat representative of the number of species that they've detected. What I thought I was kind of curious about in this area is how do the species that they detect in New York City at this array called New York City compared to what they get in sort of suburban Albany and out in a more wild Albany site called Wild Bogan. And then we've also got these interesting sites off on an island, which often islands will have fewer species. So here's one called EDG. So if you see before, you know the first step is once we've selected species richness, we have to add a new group. In this case, why won't it? We're going to pick, let's just pick New York. No, let's see. Why won't this scroll up? Sorry, it's getting a little. All species. So the first one is called New York City. So we just type in New York City. We'll pick that one and we'll save as a single group. Save group. Now it shows up here, all species New York City. We can just edit this one. And the next one is called Bogan, after Dan Bogan, who runs this one. Bogan, this one is in the Albany Pine Bush. Oh, we need to get rid of, sorry, we need to get rid of the New York City one. See, it says select none, and then Bogan. So let's do both. This is the Pine Bush. So we'll call it suburban Albany. Save group, edit. Now, Bogan, get rid of that. Get rid of the suburban one and highlight the wild Bogan. Oops. Sorry about that. I messed that up. Do it again. Wild Bogan, save group. And then one more, what's this site called? This is called EDG. So we'll just, again, it's easier to just do G. Okay, so now, oh, I need to get rid of Bogan again. See, the nice thing is it tells you what's down here. And I can see, oh, I have Wild Bogan, I need to unclick Wild Bogan. So you can, again, see, so we've got all species New York City, all species suburban Albany, all species Wild Bogan, and all species Edge, which is over on this island. Let's generate graphs, see what we get. It's telling you how many total observations each of these sites has. You can see New York City has a lot, 25,000. And this is our graph. Let's go ahead and you can see the, we've got a green, a blue. What happened to Albany? Not sure what, I think one of them didn't work. Let's see, okay. Looks like I may have added the same one twice. So anyway, we can see here that we've got all species on the island, only 5 species. That's not very many. New York City, up to 13. And the Albany one, now I think I added it twice, it's Wild Bogan, is up to 21 species. And importantly, notice that we've plotted this on camera days. So that's how much effort has a study spent surveying mammals, because generally, as you keep running cameras, you'll find more species, right? Your species accumulation curve, that's what these are called, goes up. But we can see the island site and the New York City site, these have really plateaued, right? They got one species recently, but generally they're not adding species very quickly, so we know this is probably around the right number. Whereas the Bogan sites are still going up, so they might still get more species. They may not have reached the plateau yet. So that's important to look at. Now we can also, as in the other ones, we can view and download the data table, right? So this is how many species were detected each day from that study if you want to recreate the species accumulation curves. And you can even see the actual species data. If you're wondering like, well, what were they actually detecting on that island or in New York City? You can see which species they detected on which day. This would be a great thing if you wanted to make a Venn diagram as well to see which species were seen in different places. So these data are readily available to you. Okay, so hopefully you get the idea from that on general species richness. I'm just going to show one more exercise that I thought was pretty fun when was playing with this data, and that's to do sort of broader scale comparisons to look at broader scale patterns, not just one array versus another array, but let's add a new group. We're going to stick with all mammals, and let's just go up here in the Northwest. Let's go to Washington, select all sites, all species Washington, save. Okay, now let's do the same thing, but now let's go down the coast to Oregon, collect all species Oregon, edit, next. Let's do California, select all arrays, and let's do, just for kicks, let's do Arizona, select all arrays, save group, generate graphs. Okay, so now it's taking for the west coast of the United States, north to south, and looking at how many species were observed, combining all the cameras together from those states. So we know California is a bigger state than the other one, so it might have more species. It's also a little bit south. So here, Washington, the furthest north, has the fewest species, right? And the nice thing here is you can also combine at the same effort, right? At 300 camera days, Washington had fewer species, followed by Oregon, followed by Arizona, and California, very similar, even though California is bigger. So this is kind of a fun way, I think, to look at species diversity patterns at broader scales. And again, it'll color it on the map. Yeah, so I think that's all I wanted to do for species diversity. I hope you enjoyed this and will find some cool patterns yourself. I look forward to hearing about them, hopefully at the Snapshot USA Student Research Symposium. Or if you guys want to, publish the data from this, you're totally welcome to. So get after it.